Alright, so we're moving on with asexual cell division, and today we're going to primarily focus on mitosis. So when you think about asexual cell division in general isn't just mitosis. Cell division could be mitosis, but it is also meiosis. And what we're really going to try to focus on is the mechanism of asexual reproduction or of mitosis. So we're going to see what happens, why does it have to happen, how does it happen. So first question we want to explore is, what is cell division? Cell division is simply taking an original cell that's often known as the parent cell and dividing or making copies of it. So you have an original cell, but first you need to copy the DNA. Then you need to divide the copies of the DNA, and then eventually you'll divide the cell into what's known as daughter cells, and that's going to be your final product. But for a cell to divide, for cell division to occur, you must have a signal to get started. All right. The division of the cell or of the nucleus, mitosis, is only a small part of the cell cycle. So if you remembered when you had a picture of the cell cycle, for most of that cycle, and I'm going to represent that by green, most of that cycle, the green is going to represent interphase, which was the normal life of the cell. It was where the cell divided its DNA or made copies of its DNA. So that was G1, growth, S, replicating DNA, and G2, which was more additional growth. But there's only going to be a small portion of the cell cycle where you actually divide the cell or go through mitosis. So you'll have mitosis and cytokinesis occurring. That's a very small portion of that cell cycle. If you continue to, and actually if you would take a minute and look at the cells that are above here, here in this picture you're actually seeing plant cells, and specifically this cell right here is a regular plant cell. But right beside it, this cell is a little different. This cell is irregular in size and will actually need to divide soon. So it actually needs to divide soon. So if you notice the difference there, it's reducing that surface area to volume ratio, so we're going to get a signal pretty soon and needs to split. But moving along, so why do we want asexual cell division to occur? And we really do want to get comfortable with the idea of the why and the how, the details of this process. So why do we want asexual cell division? To make or produce clones or identical cells. And this is the idea of growth or repair. But in asexual cell division, we have a lot going on in this fairly simple picture. So the first cell we have here is known as our parent cell. And in our parent cell, we can actually see that there are some homologous chromosomes. Hopefully you'll remember that homologous chromosomes are chromosomes that have the same information in them, and so they do come in pairs. So in this particular cell, we have 2 chromosomes, and that only gives us 1 homologous pair. When we talk about this number 2, and we did review this a little earlier, when we talk about the number 2, this is going to be our total chromosome number. And this number is referred to as the diploid number. And the pair that we have or diploid you can think of as having the total number or pairs. Okay, that's what diploid means. The cell right now has some single homologous chromosomes. The cell is going to go through DNA replication, and DNA replication is going to happen during the S phase of interphase. When DNA replication happens, we still have only 2 pairs of chromosomes, or 1 pair of chromosomes, but 2 homologous chromosomes. So this here, we still have 2 chromosomes. 2 chromosomes at the first cell, 2 chromosomes in the second cell, except now they're doubled. Remember that the cell is going to have to get a signal from here to here in order to actually replicate those chromosomes. When we're done replicating our DNA, we are going to move on to mitosis. And mitosis is specifically the division of the nucleus. And all that's happening is that our initial chromosomes that were singles, or singled, have doubled, and once they have doubled, they will be single again at the end of mitosis. So notice that we have 2 chromosomes, they're going to be a homologous pair, and they're going to be individual nuclei, and they're going to be single again. From there, this one cell is going to go through cytokinesis. And in cytokinesis, it's going to produce 2 diploid cells. Still has the whole number of chromosomes that are identical. Okay. And these are going to be known as your daughter cells. So this gives you an overall picture of how cell division occurs. First, you're in interphase. In interphase, your DNA replicates. After that, we get another signal, and that signal is going to tell us, okay, now we can divide the nucleus. Once we divide the nucleus, we're going to make sure that this has the correct amount of chromosomes, and then from there, we'll split the cytoplasm of the cell to produce 2 new cells. So now we need to look at the stages of mitosis, okay? In the stages of mitosis, we're going to start with interphase. So in this first cell, we're going to be in interphase. And one thing you want to keep in mind, when it says what are the stages of mitosis, these stages are continuous, meaning they are not completely separate from each other. So our first phase is known as interphase. And in interphase, you're going to see a couple of things. In interphase, we'll be able to see some relaxed chromatin. And relaxed chromatin just means uncoiled DNA. You're also going to be able to see that we do have another structure inside the cell called the centriole. And the centriole is going to coordinate spindle fibers. You've learned in Bio 1 that centrioles are only found in animal cells, but we're going to change that now to mainly in animals, because new discoveries are always happening, and that's not necessarily 100% true each time. So in interphase, we're going to have a couple things happen. Again, we need to just keep in mind that interphase is going to be G1, growth of the cell, S, replication of DNA, and G2, additional growth of cytoplasm and organelles. Then our cell is going to receive a signal and be able to move on. When it receives a signal, the next phase that occurs is going to be called prophase. In prophase, you'll see that the spindle fibers will form, and the spindle fibers are actually made of a cytoskeleton protein. They're actually part of that protein that makes up the cytoskeleton. Notice that you're also going to have, one of the main things that happens in prophase is that your chromosomes become visible. Okay, chromosomes have become visible, and now they're actually also doubled. Under a microscope, you'll literally be able to see them now. Whereas in interphase, you would just see a dark mass here, or dark blob here. Then, what phase is next? Hopefully you remember that that's going to be, you probably think you metaphase, which is what I started to write, but it's actually pro-metaphase. And in pro-metaphase, what occurs is that our spindles attach to kinetochore, your kinetochore protein on the chromosome. So there's actually a protein that's attached here on the chromosome, and your spindle fibers will attach to the protein, not directly to the DNA. And the other thing that happens is that your nucleus is going to break down. This is pro-metaphase. Spindles attach, nucleus starts to break down or is broken down, and then we're actually able to move to the next stage, which is metaphase. So in metaphase, very simply, spindles move the chromosomes to the middle of the cell, which is also known as the equator. Okay? Here, we will actually learn that there is a special metaphase checkpoint that's going to help regulate this process. We'll continue moving on, and we'll move into anaphase. And in anaphase, what's going to happen is that our sister chromatids, the copies of DNA, will be separated or pulled to opposite poles. Okay? So opposite ends of the cell. So this and this are identical copies of each other, and they're going to be pulled to opposite ends. And then we have the last phase, and in the last phase, this is known as telophase. And in telophase, the nucleus will reform, and mitosis, not cell division, is complete. And each cell has identical copies of the DNA. Okay. And we'll actually finish up the rest of your notes in class. Don't forget to do your note card, and it's actually about this kinetic core structure.